All right, folks. Well, it's Healthcare Unfiltered. I'm very excited because, first of all, I have a new guest that I've admired through Twitter and through interacting with him on social media. But it's the first time that he comes on the show. I'm very excited to have Dr. Ross Levine. He'll introduce himself. And I have two recurrent guests, Alan Skarbnik, who goes by Scarbs. You know, anytime you have like a nickname beyond your name, you know you've made it. You know you've made it. And I'm hoping to be called CN or something. And then Dr. David Steensma. So let's do a quick introductions, and then we will get started and talk about three main topics: career paths, mentorship, and medical meetings in the time of COVID. Ross, get us started. Tell listeners and viewers a little bit about you.
Thanks. Thanks for having me, Chadi. I'm a physician scientist at Sloan Kettering and a leukemia doctor by training. And I actually did my formative training at Dana-Farber, which David knows well, and then got to Sloan about 15 years ago. And I've run a lab here for the last 15 years studying leukemia genetics and biology and therapy, always at sort of the interface between the lab and the clinic, working with lots of great folks here and everywhere else. Two other things. I now, I guess, have a larger role here overseeing all of translational research at Sloan, which has been an interesting career transition in the past year for me. And I've had the amazing experience of having just the most spectacular people and colleagues in my career. And that does include both Dave and I have had many amazing interactions over the years. And Scarbs was a trainee in my lab in our formative early years, which maybe we could talk a little bit about, because it was, I think, very useful for both him and for me and for the whole group.
Absolutely. And Ross, do you have a clinical responsibility? You see patients or just mainly in the lab?
So I until 2022, I always attended on our leukemia service for 4 to 6 weeks a year. And then this past year, I decided to pivot, and I'm now just doing a small, as David will appreciate, clonal hematopoiesis clinic. But I do have a small continuity clinic, and I've sort of pivoted from inpatient only for 15 years to now outpatient only, at least for the moment. But yeah.
So you're going to tell us a little bit of the pros, cons, and the pains and aches of being a clinician scientist or a science-based researcher, because I'll tell you my view that how much guys you struggle for funding and lab and all that stuff. Scarbs, welcome back to the show. You know, I still maintain that one day me and you are going to have a restaurant together, judged by all of the posts about your barbecue. But a little bit about you, and you're representing the career path of a clinician researcher.
I am. Okay, that's news to me. But first of all, thank you for inviting me. Unfortunately, I won't have a lot of time here. I had a different commitment, but thanks for thinking of me. Thanks, Ross, for thinking of me. It's a pleasure to be here again, and certainly we'll open our restaurant someday. Skarbnik, I am from Brazil originally. I went to med school there. I did my internal medicine residency there. And during that time, I had the opportunity to visit some of the cancer centers in the US. I spent some time at MD Anderson, a couple months at Dana-Farber, where it ended that the comparison between, at that time, the training that was available here for oncology, hematology was vastly different than would be available in Brazil. And I thought it would be a good opportunity to come over. So I came over here in 2007 to redo my internal medicine residency in New York. I did it at St. Luke's Rosewood, which was a community hospital affiliated with Columbia at the time. But what we did at the time is we covered the floors, the infirmary floors at Memorial Sloan Kettering, where I met Ross in the leukemia service. And at that time, I was particularly interested in leukemia. And I asked Ross actually to give me an opportunity to kind of come to his lab and see what's all about and try to learn and see that's what I want to do. Ross was extremely generous with opening the doors for that and with his time. And helped me to, I have never worked in a lab before. That was truly the first time that I pipetted something since med school. But, you know, I learned a lot from Ross, from being the lab, from I think it was the first or second year of your lab at that time.
Yep.
It was very early on. I almost set his lab on fire. I believe.
You did?
Almost. Almost. He caught on fire.
There's a young Dr. Abdel Wahab, who David knows well, in the lab at that time, who was just starting his training as well.
Omar was there, and it was a great experience. I had a lot of field experiments there, but it was an important teaching to learn to deal with failures, actually. So that was very cool, and Ross was tremendously helpful in opening the doors for me afterwards to help me get a fellowship, wrote an awesome recommendation letter. So it was very good in that sense, and I'm really tremendously thankful to you up to this day for all of that. And then I did my fellowship at Fox Chase Cancer Center, and then I did my transplant fellowship at City of Hope. And then since then, I took more of a clinical pathway rather than a lab path. I mean, it was important for me to know that that's not what I wanted to do, too. So I think having that experience and leaning in a different way was important. So I've been working with transplant cell therapy and mostly lymphoma and CLL since then, but I focused on clinical research. So that's it in a gist.
Thanks, Carbon. And I know that you're tight on time. We're very appreciative, but we thought this is a great mentor-mentee type of thing.
Absolutely, yeah. And I have to add that, even since I left, Ross is still very generous. Every time there was a meeting at ASH for his lab, he always invited me. I always participate, even though I was there for a short period of time. I still felt a part of it. So that was
and
great.
You're from Brazil, so Doha 2022. Let's see what happens, right?
I don't have much faith in my national team, but we'll see. Yeah. As long as Argentina doesn't win, I'm happy.
David Steensma, tell us a little bit about what you are career-wise right now.
Sure. I am academic at heart. Until 2020, I was at Dana-Farber, previously at Mayo Clinic, had been a University of Chicago medical student. And in 2020 was recruited to NIBR, which is Novartis's early drug development institute, like a biotechnology company within a larger pharmaceutical company. Recruited by Jay Bradner, a very dynamic scientist, stem cell transplanter by training. I had known Jay from Dana-Farber, where he was for a number of years, focused on chemistry and drug development, and he was a stem cell transplanter. I was trained as a MDS leukemia doctor and had a heavy clinical practice of MDS marrow failure. And then in the last 7-8 years, when I was seeing patients, it was really focused on still very much MDS, but started up a clonal hematopoiesis clinic very shortly after Ross and Kelly Bolton and their colleagues at MSKCC started one. That was a fascinating experience, and I really enjoyed that. What I do now is I lead hematology drug development for Novartis, at least early drug development. And that includes everything from talking with the scientists who are working on target discovery, on lead candidate optimization, on GLP toxicology studies, to supervising physician-led teams who are designing the first-in-human trials, interacting with regulatory agencies, and it's all across hematology. So everything from sickle cell gene therapy to novel CAR Ts and everything in between, heme malignancies and non-malignant teams. So it's been a really dynamic, fun. I've been doing this about 20 months now. And it's really been very cool. I still get to interact with a lot of investigators. It's a really cool community, the heme and heme malignancies community. And I think, my friendship with Ross really exemplifies that. I met Ross probably 10 years ago now or more. More, I think, actually.
We wrote our first paper together before we ever met in person.
That's right, we did do. Yeah, back when Gary Gilliland was still at
2005.
Yeah. Wow, 17 years. I've just benefited immensely from contact with him. He's absolutely one of the most smart people I know. Every time I talk to him, I learn something. So, and then meeting you, Chadi, in this context, we first met when I was a JCO section editor, and you had submitted a manuscript, and we worked through that got published. And then I was on this podcast once before with Mikhail Sekris, whose career in some ways has paralleled mine. Mikhail is now the chair down at University of Miami, and he left Cleveland Clinic for a new role the same time that I left for this new role in Cambridge, Massachusetts. So that was our previous Healthcare Unfiltered podcast.
We talked
so I already have one t-shirt.
Yeah, well, this is your fourth appearance. We talked actually the other time when you wrote about the Raven in JC.
Oh, that's right.
I was very.
Oh yeah.
Forgot about that. That was right at the beginning.
I was very intrigued. I was an amateur podcaster. Look at me, and I'm a professional. But
come a long way, baby.
But the episode with Mikhail and you was really more focused on career change. Like we talked about what career change. What I want to focus a little bit more, what happens in the days in the life of that career. What do you do every day in that career, and then segue into additional topics. So Ross, I want to start with you. So if I'm listening to this, and so you're a lab-based scientist and so on, in addition to what got you into this, because I presume when you go to medical school, few people know that they're going to be lab-based in general. When you first go to medical school, into the rotation, you're really thinking you're going to be a doctor, a clinician, and taking care of patients. Not that you're not, but you rarely think right away you're going to be a lab-based scientist. So in addition to telling us how this happened, what do you do day in and day out? How is your day or week structured as running a lab?
Yeah, so two great questions. I'll try and address them one after the other, because actually the story of how I became a scientist at least got some interesting components of it. I'm an MD, I don't have a PhD. When I was an undergrad, I didn't have any really laboratory experience, kind of like what Alan said when he showed up to my lab. I was not either. I had colleagues, including Jay Bradner, who was my college classmate, who was in a lab, Mark Patashi's lab as an undergrad at Harvard. I didn't work in the lab at all. And so I got to medical school thinking exactly as you said, Chadi, I was going to be a clinical expert in something, and I was excited to figure out that was. But what happened to me was that when I got to med school, I had a interaction with a guy named Bob Kerman. He was my father's colleague from college. My dad's a retired dentist, but they went to undergrad together. And he was at the time a division head at Johns Hopkins in gynecologic path. Scarbs, be well, my friend. Miss you, ma'am.
You too. Thank you for the invite. It's a pleasure to see you all.
Having you for 5 minutes is okay. You probably can log in back. We'll probably be still talking in the next hour.
Yeah.
So.
Yeah. Hopefully I'll touch base. Sorry to interrupt.
All right.
Thanks so much.
Yeah.
Bye.
So I didn't know what I wanted to really think about and do. And I met this gentleman who was important in my career. He was a pathologist, no molecular background, but he was the only person I knew at the med school. And he was someone my dad knew. And for the first month of med school, I never went and saw him, because when your dad tells you to go talk to somebody, the first thing you do is not, when you're 23, listen to your parents. But after a month, I was like, you know what, maybe I should call him up. So I went to visit him, and he was in his office with his 6-headed scope. And he's like, the future is molecular biology, and you need to go do science. And the same way that some people, if you remember the story from The Graduate of math being told to work, Dustin Hoffman to work on plastics. It was that same story. Science, you should do science. And so he put me in a lab basically by calling up 2 junior faculty, amazing women, Laura Hedrick Ellenson, who's now actually at Sloan, which is great because we get to interact. The other was Kathy Cho, who's is now at University of Michigan. And they had just started their labs. They'd done their postdocs with Bert Vogelstein, who's an iconic figure in cancer research. And I got to the lab, and like Scarb said, I was like all thumbs, right? Nothing worked. But that first summer, I worked in their lab and nothing worked. And then my last week in that summer, all of a sudden I found stuff. I found mutations in a cancer, in this case, serous uterine cancer, had never been found. And it was the first inkling that we would have a publication that ultimately a postdoc wrote, but I like got to see discovery, something I found something with my, at that point, hands and eyes that no one had ever seen before. And I could go into my PI's office and go, I discovered mutations in this gene that had never been seen before. And it was like, I was hooked. I was like, oh my God, like this is the moment. And I think the other thing was that I also realized that science was going to impact medicine. And I was like, this was going to blend well with an interest in medicine and human health. I did a gap year between my third and fourth year med school. I was at Johns Hopkins. It was amazing. I remember leaving the clinic and going to the lab, and I wasn't like, oh my God, why did I do this? I loved the lab. I loved the clinic. I always found the back and forth very seamless. I went back to the wards after my year. I loved, great experience in the lab. And then I went to fellowship, residency, and then fellowship. And I was like, I'm going to do this clinician scientist thing. I'm going to be that physician scientist. And I remember I wrote my essay for fellowship where I was going to do that. And so I did a year at the Farber. I found I loved leukemia. I knew that that was the area I wanted to do. I had amazing experiences on the boards, but I felt like the science was the problem with leukemia, that the drugs weren't there. And that if I was going to focus on leukemia in 2002, 2003, I had to be in a lab and figure it out, because there was just no exciting, there was just no targeted therapy. It was a tough time, as David will know. And so I met a guy named Gary Gilliland when he was giving a talk to the first year fellows. And he said, targeted therapy is going to change the world. Gleevec has just come out. It's this great moment to find mutant genes. And I'm like, I'm going to work with this guy. And I went and I called him up, and he's like, you got to come to my lab. And the last part of the story I'll tell, and this is important, I tell this to the trainees all the time, was that I was told by, at the time, my fellowship director, I should be careful because I don't know if I've ever said this publicly, I've said many things, that I should never be a scientist, that I was destined to be a clinician. I didn't have a PhD, and that I should really think long and hard before I went in the lab. And probably the most important thing I did was not listen to that advice and go to the lab anyway, not because it wasn't good advice, but because sometimes the best thing you do is take good advice, but if you believe in something, do what you want anyway. But it did cause me to think about it and really commit, and I committed. And I was in Gary Gilliland's lab for 4 or 5 years. I had an amazing experience. I was lucky because we discovered the JAK2 mutation when that was my postdoctoral project. And that really obviously was a high impact discovery and gave me an opportunity to run my lab. And I came to Sloan after that in 2007 when Memorial started its new translational program. I've been there ever since. And so that's my story, and I'm happy to delve into it. And like I said, David and I wrote a paper even before we met, which was our second JAK2 paper. I think it was a blood paper, right, David? Yep. And I think you're first author, and I'm second, with Teferi and Gary on the frequency of JAK mutations across myeloid malignancies. Yeah. But you know, my day now, I always say that
before you get to your
go ahead.
Before you go to your day, Ross, one of the things you mentioned is pretty interesting, and maybe you could elaborate a little bit on that, because I think from a listener perspective, it's probably gonna resonate with them. You're an MD, not a PhD, or not an MD PhD, and there is a sense, maybe a perception, maybe wrong, maybe right, because PhDs are trained differently. MDs might have a little bit more of a challenge to really be in the lab, completely focused in the lab and running a lab. Is this a perception or reality? Did you say?
I think it's reality. I think there are some real headwinds for MDs who want to devote their career to predominantly laboratory science. One is that you get typecast, and I think that was my experience, that it's easy to not think that someone who doesn't have a PhD, that they're going to go to the lab just to try it and they're not going to stick with it. That's one thing. I think there are a lot of training programs in oncology where you only get a year and a half in the lab, and even during that year and a half, you're always being pulled to clinic. If you're an MD and you're going to do science, like I needed 4 or 5 years of dedicated scientific time. And I had a program and an institution and a mentor who worked hard to protect my time. It did mean, for example, that I deferred joining the faculty for at least 1 extra year. And so you got to make some choices. And Erica,
so you did this 4 years after fellowship, or
I did a 4-year hem-onc fellowship, which included about 2.5 years of dedicated research time, and then tacked on 2 more as an instructor at the Farber with very limited limited clinical involvement. So I really had dedicated, protected time. They put me on training grants. I wrote career development awards, but most importantly, people believed in me and thought that I needed that time. And I think it's really hard to, in 2022, unless you're at a very small number of institutions with the right mentors, to make sure that people get enough of a sort of cocoon. And I don't know what, David, you think. I worry greatly about it. And what I've noticed in our sort of faculty applications we get for independent is the drop in the number of straight MDs who apply for faculty positions, which means many of them aren't even getting there. And I hate to use the word that they're falling off the tree. That's not fair. I think there are great paths, and being a clinical investigator and having been in the lab is also wonderful. But we're worried that the number of MD only folks who go into science are limited. Craig Thompson, who's our outgoing CEO at Sloan Kettering, is an MD without a PhD. Charles Sawyers, who's been my mentor and boss at Sloan, is an MD. Harold Varmus is an MD without a PhD. But I'm very worried that that sort of path is getting harder, and it needs more support than we're giving it.
What do you think, David?
I agree. Do you want me to talk to how I got to where I am, or
Yeah, you could do that, and then I'm going to go back to Ross about his day-to-day activities. So we'll go back and forth this way.
I think what Ross says is very true. And I think it's changed. It was never easy for MDs only to start a lab career, but it's become more difficult in recent years. And part of that's because clinical medicine has become increasingly burdensome. You're spending a lot more time documenting, you're spending a lot more time doing prior authorizations. When you're young, you often have less help and protection to do that. And so unless you have a really special microenvironment, and those still exist, but they're far fewer than there were, is my perception. It's really, really hard to do. So I went to medical school having thought at first that I was going to do graduate studies in astronomy and publish some physics papers, and that was the direction I was going. And I took a cell biology class just for fun in college because I'd already completed my major requirements, and I thought, wow, this is really cool. Maybe I should, take organic chemistry, which was the only thing I didn't have that I needed for med school, and go to med school instead. So I took a year's worth of organic chemistry over the summer between my junior and senior years, and the rest was, history. But I went to medical school thinking not necessarily that I was going to do science. I was certainly open to the idea. I had some really good mentors, Jim Vardaman, Janet Rowley, John Altman was very influential at University of Chicago. And, I met Dick Larson there, also, great role model, and ended up gravitating towards hematology. Went out to Mayo Clinic because my wife we got married during med school. She was doing a PhD in English at UIC, and she'd had enough of living in the big city and having our car broken into, and my classmates were getting mugged all the time, and it was just
it's Hyde Park. It's Hyde Park. It's not
well, yeah, it's not so easy. But so we went to Rochester, Minnesota, which turned out to be great. I loved Mayo. The winters are tough, of course, but so many good people there. Mayo is a very clinically heavy institution, I will say. I got great clinical training. I went into the lab as an elective my first year and just thought it was super cool. Made a little tiny discovery in red cell biology, and I thought, wow, I could do this as part of my career. And so, but like Ross, I realized without a PhD, I was at a disadvantage. I stayed at Mayo for fellowship. But in order to get some extra time preparing for the lab component, I went away to the UK for 2 years, got a visiting scholar. And there I didn't see patients except, tagging along a couple times a month with a consultant. So I was in Oxford at the Weatherall Institute of Molecular Medicine, studying globin gene regulation in the context of myeloid neoplasia. It was terrific. We discovered a new somatic mutation. And then I came back on the faculty in 2004 with a K award and a lab of my own. It was a small lab, but it was fun. And I had gotten the idea when I was in Oxford that there were likely to be spliceosome mutations in MDS, and started looking at that, got distracted by some other projects. That was a little bit of a challenge. And I, kept applying for funding to try to study this spliceosome and MDS, started looking at some candidate genes, picked the wrong ones. And eventually, I kind of faced a choice where I was doing more clinical trials, a lot of investigator-initiated trials, because pharma wasn't interested in MDS or MPN at that time. So they were all IITs. Ilu Teferi was a clinical mentor around myeloid disorders and clinical trials. And 2008 came around, and I kind of had to decide, because the K was coming to an end, what should I do next? I got some great philanthropic funding, so I could have kept it going. But Rich Stone called and said, gosh, we really need somebody, out at Dana-Farber. It's a much more clinical position. You can work with Gary Gilliland if you want, but it's going to be really hard because we're going to work you really hard on the leukemia wards. And so I thought about it, talked to my wife about it, and went, and and got there. No sooner had I arrived than Gary left. But there was a terrific young investigator named Ben Ebert who took over a lot of Gary's lab space. Ben was a great collaborator. And so that was a lot of fun. And then, was really in a good position. I was well funded, actually, enjoying what I was doing, had a good mix of the patients I wanted to see, great colleagues, but, Jay Bradner's a persuasive guy. And when he called, I initially said no. Industry doesn't seem like the life for me, but he called again, and I came out and visited, and one thing led to another. And, it was 2022 also, and I was turning 50 in that December. So it's one of those life milestones where you think, what do I want to do if the creator gives me another 15, 20 years?
Usually you buy a
life,
top-down Corvette, and that's what you do.
Yeah.
Well, no, that's not my style. So I thought, maybe I should try this and see if I can influence health in a different way. And that's how this came about.
You remind me of one of my colleagues at University of Chicago. I'll mention him. His name is Walt Stadler. Hopefully he'll be listening to this at some point. He used to tell me, no is the start of the conversation. So when you said no to Jay, he's just gonna keep going again. So Ross, okay, tell us how your day looks and your week looks. What do you do every day as a lab scientist?
Maybe for fun, what I'll do is I'll tell you what I did today. How about that?
Yeah,
it's a pretty typical day. After I went out for my early run in Central Park, I met at about 7:45 a.m. with the chair of medicine, Deb Schrag, who David knows well, because we were trying to figure out some areas where translational medicine and the Department of Medicine, where things we can figure out together, faculty recruitment, strategic areas, biomarkers. And it's been hard for us to find time during the day. And so we thought the simplest thing was to meet for breakfast because we both have to eat. So we met for breakfast. At 9 a.m., I did a Zoom with an investigator in Italy who is from the ASH TRTH program, who wanted some advice on a proposal because I was on TRTH this year. And then let me look back. I had almost all of it memorized. I then met with one of the postdocs in my lab, who's a pathology faculty member who works in my lab on MRT and RUNX1, and we sat in my office for a half hour. We went over data. And then I had about an hour to sort of answer email and write and think. And then at 11 o'clock, we had lab meeting from 11 to 12 in person. And one of the newer postdocs, who's a physician scientist in my lab, presented exciting new project where he's making a whole bunch of new mice. And we got to sit and dig into it as a group. And then at noon, I actually had a meeting with a company I founded with some of the folks who were involved in helping me do that, because we had some decisions to make about what we're doing. And then I had a couple more meetings with lab members. I met with one of my postdocs who's from Spain, whose project's at a crossroads, and I actually asked another lab member and the technician he works with to join so we could make some choices and whiteboard some experiments or ideas. So we did that. And then I had a Zoom meeting with some colleagues at AstraZeneca who just wanted my advice on something. Anas Yunus, who you guys may know well, and one of his colleagues. So we caught up on some things. We just wanted to chat. just to catch up. And then I had a 4 o'clock meeting with one of our with the head of our research administrator, because we're looking at our budget for next year, and we're trying to make decisions about administrative support and how we can be more efficient and effective. And then I met with one of my grad students who is about to do her thesis qualifying exams. She's an MD PhD. She's been in the lab about a year, and she has to now have her proposal evaluated by her committee. And so we were going over her ideas and aims and getting those into shape so she can show her committee. And then after a little bit more email, I came home. I walked a block from Sloan to my apartment where I am now, and I got on the podcast. So I don't know if that gives you a flavor for my day. It was Zoom. It was sitting down with people with data. It was talking to colleagues in academia and industry.
It does. So I think I'm gonna subclassify your usual days is a lot of meetings.
Yep.
These meetings could be internal meetings with, and some of them are external meetings. And then you have the lab meetings, which is it daily lab meeting you have, or once a week?
We as a group, I do a weekly lab meeting with the whole lab, and then I meet with each person in the lab an average of every 2 weeks. But many of those people have a weekly slot, and we just sometimes that we decide everything's good. I actually think, I always say the most important thing you do as a mentor is when your lab trainee says, you know what, I don't actually need to meet with this week. We met last week. I'm good. I just need the time to do experiments. I'm like, I'm always happy for you to give me my time back. And then you get your time back.
So the type of meetings, internal meetings, external meetings, could be with collaborators, mentees, or industry.
Yep.
And then there are the lab meetings piece. Are you ever involved at this stage of the game with, spending time in the lab, watching the technical aspect of an experiment? Like, is this something that you just did when you were a junior faculty and no longer do, or are you still involved in that?
So everyone runs their lab differently. I probably for the last 10 years have not physically done experiments. I have a lab of 18 people, and I think that it's more important for me to sit with their fresh data and help them think through it than me to actually do experiments. I'm in the lab, I would say on a normal day, 3 to 6 times a day in the physical lab, because I'll just go in and talk to people. I'd be like, what's up? Let's talk about this. Or I had an idea. And I think that, especially with the pandemic, where there's less interaction with the outside world, I've actually gotten more interaction within me and my lab. In fact, for most of the last 2 years, the only people I see in person are my lab members. Almost everything else is by Zoom. So that's changed a little bit as we do more meetings. And actually for me, that's been enriching, because when pre-pandemic, we'll get to meetings, when I was often traveling and electronically with my lab, I actually find the flip more fun. I would rather spend more time with the people who are in my group who are generating data in person. To me, that's the part I think that gets me the most excited, is when somebody in the lab's got a new result and I can sit with them and think through it. That's the part that even as I take on leadership roles, I can't live, at least for now, without.
Okay. But when you first started, I presume the first couple of years,
I was in the bench. In the bench, and what I did, every lab is different. How I did it was that I took the first 4 or 5 people as they started the lab, including Omar Abdel Wahab and Priya Kapakar and others, is that I sat with them and I really sort of worked alongside them and got them going and helped them do things, and that was great. And it meant that in many ways, like, we were side by side, but within a year or two, they were sort of like, Ross, we can do the experiments. What we need your help in is how do we figure out what those results mean? How do we get it into a story? How do we get the papers and grants? And that's a style thing. There are other people that spend their life always at the bench, and I have great admiration for that. I don't think how I do it's the right way. It's just how I do it.
No, as I listen, to me, it seems it's the logical way. As you progress, you want to help differently. They will need more help thinking as opposed just to the technical stuff.
But if you had a lab of 4 or 5 people, some people that keep their lab small so that they can work at the bench next to their people, I think that's a wonderful model too. But anyway.
All right. So David, you had a question for Ross, and then you're going to pivot from that to tell us a little bit about how your day-to-day looks like.
Yeah. So my question for Russ is, at this stage of your career, there's a lot of people who have come through your labs who have now set up their own independent groups, or they've moved on to become, faculty members, some of them quite clinical at different institutions, others, with a heavy research. Do people still reach out to you regularly for advice once they've moved on to new institutions, and what's that been like? Because some of them have stayed at MSKCC, but you've also now trained a lot of people who have set up their own groups in different places.
I don't know. I keep in touch with everybody, pretty much, who's ever been through the group. I just gave comments on an R01 to one of my former trainees who's at Mount Sinai. I've got somebody I trained with who's at Columbia, Aaron Vinny, who you know well, who I've talked to almost every day by email or by Slack. Nicole Cousine, who went across the street to Cornell and does pediatric MPN, I still meet with her monthly. And my attitude is that my lab is a family. And whether you're in the lab or gone, you're part of it. I also think, to be blunt, and this is something people don't appreciate enough, I actually feel like I get a lot of mentorship from my current and former lab members too, about science and my choices. And I find that bilateral only gets better as people move on. I think Gary taught me that when I was in Gary Gilliland's lab, I think I felt like I was sucking, all the juice out of the orange that was Gary's wisdom. But when I left, and then when he went to Merck, he and I would still meet every year, and I felt like, although it was never truly symmetric, it was bilateral. And I think for me, that was an important thing. I will tell you that when I have choices to make about my career, my current and former group are the people I tend to go to first and foremost. And they're incredibly helpful to me, as I hope I am to them.
Thank you.
Very good point. I usually call this the advisory council that each one of us have, that they may not be always visible, but that these are the ones you will call when you're trying to make an important decision. David, what's your day looking like?
Yeah, my day is really varied, and I like that. There's a lot of time. Increasingly, we are going into the lab in the office. However, the majority of our meetings are still on MS Teams. The range of meetings is really quite wide. So they can be anything from meeting with scientific investigators, and they may have a new target that they want my opinion on, or they may have identified a couple of different development candidates, but each one has a different liability. So they say, in this patient population, is it more important to have no QT prolongation, or is it more important to have something that can be given once a day instead of twice a day? And those types of meetings. There's also a lot of just more theoretical, where they say, what if we could do this? We think we might be able to degrade this protein. Would this be useful? We think we might be able to make a tri-specific antibody with this co-stimulatory molecule. What do you think about that? Then there's a fair number of meetings with the clinical groups. So there's 5 clinical program leaders, physicians, who are all terrific, who I'm their manager. Each of them has 3 or 4 different programs that they're leading. And so I meet with them at least once every 1 to 2 weeks, sometimes more frequently if issues arise. And we talk about their studies. We talk about, what the recent FDA feedback was, how it's accruing, what toxicities are being seen, all the kinds of things that you would expect. What the future of the program is, particularly after major meetings like ASCO, ASH, EHA, when new data is presented, we get a lot of competitive intelligence. What are other companies doing? Are we still competitive, or are we falling behind? We had a BCMA directed molecule, but after some of our competitors have had huge success and achieved regulatory approval, we decided it just is a very heavily subscribed target, at least that one molecule. So yeah, there's also a lot of more general strategy meetings, including with our late development group. So yesterday I was in New Jersey and spent the day with our global development group. They're the people who do the regulatory phase 3 trials, the ones that are going for registration. And so once we in NIBR have developed a new medicine, taken it through first of human studies, achieved what's called proof of concept, and then we say, hey, this is what we would recommend as your go forward dose, then they take it forward for that last important critical step for a randomized study, usually for regulatory approval. I also interact a lot with external investigators. Sometimes this is around academic collaborations. We have collaborations with sites all over the world around either investigator-initiated trials, or they're doing work related to one of our compounds. And then one of the things that's still fun is I still get to do some academic type things. So, 2 weeks ago, I was at Columbia University for the day, meeting with Aaron and giving a talk there. And, it was a very quick trip, but it was fun to see some folks I hadn't seen in a while. So that's good. And perhaps leading into our next segment, one of the things, the only thing that it seemed like people in industry complain about, other than high turnover in companies and things, was the travel. And for that first 18 months, I was doing virtually no travel. In the last 2 months, that's gone from 0 to 60 miles an hour. I mean, I've had probably 9 trips in the last 8 weeks. Most of them are short, but yeah, ASCO, EHA, site visits to East Hanover, site visit to Basel, 2 of our other Novartis sites. This retreat, a different retreat in Maine, another retreat on Cape Cod for strategy and planning, visits to Columbia, visit to another academic site, ISCT out in California, IWNDS this weekend. And it's just, thankfully, this is it. Now, 2 months, there's going to be nothing, but it's just like crazy. I thought, gosh, now I know what people complain about.
I had a lot of travel over the past couple of months. I said, okay, can we get a lockdown for 3 weeks? Just 3 weeks lockdown, just please. I miss lockdown. Ross, one of the things that we hear a lot about, and many people not complain about, but it's certainly a barrier to them wanting to do a career like yours, is grant funding. Not only the process of writing grants, which I believe there's an art to it. I think I've written grants that were rejected, but there was an art in writing them at least. But what do you think is going on there? How difficult this is? How tough is to get funding? Because let's be honest. I mean, a lab career success, and correct me if I'm wrong, is really contingent on getting money. And how you get the money is going to be either from your own institution, from pharma, from the government. So tell us how, I don't know, give us the do's and do nots, and how much do you believe this is a barrier in getting more scientists into the lab?
I think it's a big barrier. And I think the model is got, inherent fundamental flaws that have existed and only get worse over time. So that's my glass half empty perspective on it. But I'm at heart an optimist. So I think, the way I've always thought of it is that you have to learn that it's, for better or for worse, sort of a system and a game. And you have to learn, one, how to do it effectively. And you're right. I really believe that, the most important thing you learn if you want to do this for a career is how to write a successful grant and what it takes to get there and how long it takes and how to handle those first 2 times that it comes back and get right back up there and revise it and do it again. And, I don't think anybody, if you design a system ab initio, would say that, most grants will need 2 or 3 rounds before they get funded, but that's the system we have. And I think that, I always say that most important thing you need to learn in academic medicine, whether you're lab or clinical, is that rejection is just a hurdle on the way to success. And, if you can't compartmentalize when your grants or papers are rejected and go home and still find joy in things and find joy the next day in data, then this is the wrong profession for you, because I think that it's really easy to dwell on the 90% failure, 10% success. And so I think my greatest asset is I just don't dwell on it. It can smart and it hurts for a moment, but ultimately you know that those are the rules of the game. On the other hand, Chadi, I think there's two things that you can do beyond I just said get thick skin. One is that I do think that some people are really good at it. And I always tell the example of Jonathan Licht, who's the cancer senator at the University of Florida, who David knows well. When I was a young investigator and I was writing a grant, one of my earliest grants was a collaborative grant, and he was part of it. And he writes grants beautifully, as good as anybody. And one, I saw how he did it. And two, he actually spent a couple hours with me on mine and said, this is how I'd rephrase it. And I learned incredible skills about how to communicate my ideas in the most effective way, in a way that the reviewer would find it visually pleasing, and that they wouldn't be like, oh my God, it's 8 pages of straight text. Like how to use, the 12 pages as a, way to, beautifully present your ideas. And I have unabashedly in my career taken great strategies from many great people. And I tell people all the time that when you work with me, we write grants together. I help you with yours, and I'll ask you to help me with mine, because then you'll learn what works for me and you'll figure out what parts of it work for you. And you can plagiarize, if you will, the strategies that work from any person and adapt them to yours. I mean, the strategy, not the text, of course. And so that's one thing I say is that this is not something that you need to figure it out yourself. There's a lot of strategies you can see. I would say the most important thing as a young investigator is get on a grant review panel and then see what grants actually do well and figure out what they did well, and then take those strategies and use them yourself. And then the other thing I tell people, and this is, I think, is that you need to diversify your funding stream. The federal government is really mercurial and hard to get money from, and there are years where they can be really brutal. There are amazing foundations like Leukemia Lymphoma Society, but then a pandemic hits and they can't run events. And then philanthropy is very capricious because a donor has to do it and decide. So what you want to do, I always tell people, it's like running a business. You need a diversified funding stream. And so I tell the lab, we're going to get from each of these buckets. And you want to be at an institution that will support you and say, oh no, we only care if you get federal money. It just doesn't work. And so I always tell people you need to build a funding strategy, because federal money is definitely the most desirable and it's renewable, but it won't pay for everything. And so I always tell people you have to think of it. Running a lab is like having a kiosk at the mall. At the end of the day, you're paying rent to somebody else and you have your own goods. But in your little thing, if you can pay your rent, you do what you want. But you do have to make sure that you have the revenue to pay your rent. And that's the way I've approached grants, is that if I can get the grants to fund my people and my space, I then have freedom, as do they, to do what we want to do. And a lot of grants will give you great latitude to follow the science and not just do. And so I view it as sort of as a means, but not the end. I don't know if that helps, Chad.
No, it does help. What do you think? What do you think are the other, what I would say, misconceptions about a lab career? If somebody is listening and just look at this, give us some of the misconceptions that you probably have seen based on mentoring many folks.
Yeah, I would say the biggest one, and there are people that strongly agree and disagree with me, but I believe strongly in the following, and this is this. I believe fundamentally that being a successful scientist and being a successful clinician or clinical investigator require largely the same logical sort of thought process and skill set. I believe that when we're in the ICU and a patient's kidneys are failing, and you think of the 19 different things it could be, you try to methodically work through all 19. You may not change one at a time, but you're looking at all 19 and you're trying to figure out how to triage and think about it. Science is the same way. When an experiment doesn't work, we think about all the different moving parts and approach it. And you have to be able to break it down into parts. But you also have to look at the pattern of results in the lab and figure out what it means, the same way we look at a patient. And I think that the thing that bothers me is when I hear clinical investigators say, I didn't learn to think like a scientist and I can't do it. Or I see PhDs who say, you're a clinician. You don't know how, I really believe a great clinician, the people I admire most, approach solving a tough clinical problem with the same thought process and logic and rigor that we do in the lab. And I think most talented clinicians and clinical investigators, if they want to do laboratory science, could actually do it. And I think this idea of getting typecast is a huge slippery slope. I don't know if that helps. I don't know what David would say about that.
Yeah, David, I'd like you to reflect a little bit on what obviously Ross said, because you also had kind of similar things of doing a few things, but also after doing that, I wanted to comment on the fact that many folks, when I see on social media or other outlets, when they talk about people in industry, they say, well, you guys have it easy. In the industry, it's easy, it's less work, it's excellent quality of life. You work much less than us in academic medicine. So tell me about that.
Yeah, that's a misperception. And I think it's also a misperception that industry is really all about, making money and trying to trick the FDA into approving your product. I mean, there are always egregious examples of particularly small biotechnology companies that get backed into a corner. They have one product and they start doing some really kind of kooky things to try to ensure their continued existence. But that's rare. And I think the people really sincerely, the people that I work with, want to make a difference, and they're in this because they want to improve human health. And sure, at the end of the day, it is a job and it is a business, but I've been very impressed by the level of discussions that we have, the level of scrutiny that all of our programs go through. Now, one of the things that I think people find a little bit difficult in industry is that sometimes you may be assigned to a different project because of a shift that's not purely science-based or not purely based on clinical results. It may be based on staffing, or maybe based on the global competitive market, that sort of thing. So I think that's kind of hard for people. One other thing that I think is hard in industry is you can be let go even though you're doing a great job. Gary Gilliland is a terrific example of that. He went from Boston, he went to Merck, and was a senior vice president there, and by all reports was doing a good job, but
developed pembrolizumab.
Yeah, I mean, a huge, tremendous impact on the health of cancer patients, right? But then a new CEO came, and the new CEO, wanted to bring in their own people and change the leaders, and so then Gary had to find a new position. Tom Lynch is another example, and his company got acquired after he'd been there a couple of years, kind of unexpected. And so, the things happen even if you are a good doctor, a good scientist doing a good job.
So, the one thing that, and this came out of conversations that Gary and I had, something he and I talked about when he was at Merck and after, is that the one thing though in academia that's hard in industry, and again, get your reaction to this, Dave, is that in industry, if you're the big boss, like you are, or Jay Bradner, if someone is multiple levels below, it is often hard for those people to speak out against and say that an idea that's already top down been bought into is actually the wrong idea. And the beauty of my job is that whatever titles I have, the grad students at the bench tells me I'm full of crap. They actually, one, will say it, and two, they get to decide and go do it anyway. You know what I'm saying? And I think that's the trick of industry, is that it's a dangerous trap to fall into if you don't figure out a way to ensure that the hierarchy, you want to make sure that that doesn't prevent sort of, people speaking out for what they believe. And I'm not saying that's cut and dry, but it's a bigger risk in industry than academia. I don't know what you think, Dave.
I agree. I think that it partly depends on the culture that you try to cultivate. We for instance, one of our CEO has this mantra he likes to use, which is an unbossed culture, and that's giving people as much autonomy. I mean, everybody of course has a manager, but I'm not telling these physicians who are the clinical program leaders what to do. I ask them what they want to do, and we discuss it. And then in turn, they're discussing with their teams. And so, if problems arise, or if somebody has something that they want to bring up, I hope that they feel comfortable bringing it up with me. But you're absolutely right. There is a little bit more of a hierarchy that sometimes is intimidating. One thing that I think has helped in that is with virtual meetings, it's less intimidating for people to put a comment in a chat sometimes than it is around a boardroom, around a table, to speak up when there's someone who's, very senior to them right there. Partly because when you're putting it in the chat, you can be a little careful about the wording and maybe think it through a little bit what you want to say. But also because I think it does level the playing field just a little bit. I think one thing that also is different about industry is just the speed at which things happen. I was reflecting on this because Novartis is going through a reorganization right now, and phase 2 was announced yesterday, and unfortunately 8,000 people, give or take a few, are going to lose their jobs, mostly in the sort of commercial marketing, strategic roles. For that to happen, and the type of reorganization that has happened, major structural changes at an academic center, would have taken many years. I was thinking it could never happen at the Farber because there's too many entrenched sort of fiefdoms and deals that have been made in the past, and it's just much more of a decentralized organization. Whereas when you have a single company, they can make these decisions where things change very quickly, for better, for worse. Yeah.
Ross, when we talk about mentorship, what does that mean to you? And maybe give me in your own words, what makes a successful mentor and a successful mentee? In the introduction for this episode, I will share a personal story where I believe, well, I'll say it here. I think some folks think they are good mentors, but they're probably not. And I think there are certain unique characteristics. So what are your thoughts? What makes a good mentor?
It's a great question. I feel like it's something I feel like I'm still learning all the time about how to do it well and better. I think for me, the most important thing is that it's about helping the mentee find their path, and not just either one making choices for them or trying to encourage them to follow your path. I think the biggest trap I see is when a mentor thinks that success is when all of their proteges make similar choices and career paths as them. I try to, as best I can, think, what are they looking for? What do they want to do? And how do I help them accomplish that? I understand it's not that simple, right? Like sometimes it's, I want that experiment to work too. And so it's not like those things are always at odds. But I do think that, helping people figure out what they want to do and then asking, what are the things that you need to get there that you need me to help? And they may not be exactly the same as what you think they are. And I have to work with you to figure out what they are. And it's taken me a long time to sort of figure out how to individualize the strategy. And then the other thing I think is that I always feel like, the things that I try to teach people are, to have fearlessness about the things they do, whether it's experimental things, like never talk yourself out of an experiment if it's a great experiment, even if it looks hard. Never talk yourself out of trying something. Encouraging people to like go for it. And to if they have fear or anxiety, helping them, understand why even if it's hard, it's worth trying. And then the other thing is self-awareness. I think people need to have a really good understanding of their interests, their hopes, their capabilities, and the things they're good at, things they're not. I always say that my most talented trainee to date is this guy, Omar Abdel Wahab, who's a friend of David's too. And he's actually more successful than I am, I think, at this stage in his career, and is a wonderful human being too. And I'm proud that I have had a role in helping him navigate that. But I will tell you that he and I have talked about this, that although he and I have similar jobs, we both work at Sloan, we both run successful labs, there are parts of what I do that he's better at than me. And there are parts of what of what he does that I'm better at than him. And he and I are able to have a pretty honest conversation that even though we're both pretty damn good at our jobs, that there's a 10% of it where I actually could use some of his help, perspective, and vice versa. That takes a lot of like honest and trust and to figure out. I don't know if those are my threads.
It takes the humbleness as well, the humility.
Yep.
But I loved what you said in the beginning, Ross, which is, because I do see it. Being a good mentor is not about everybody needs to be a lab scientist. If one of your mentees in the lab says, I want to go to industry, your role as a mentor is to help him accomplish what will make him successful in industry. And I think that sometimes is missing.
I maintain interactions with my trainees and mentees, whether they were in my lab or not, who've gone to industry. I've written papers with some of them. My strongest collaboration in the industry is because somebody in my lab went to industry and they're like, you know what, we have something now, and because I love working with you guys, and I'm like, that's the best, that's the most fun, right? Yeah. I mean, obviously we want to make an impact and you want to do collaborations that are impactful, but doing with people that you have a shared story with is something special.
Absolutely. David, is there a mentorship part in industry? In academia, it's kind of pretty straightforward. You mentor postgrads, residents, fellows, students. In industry, is there such a thing as mentorship and mentor-mentee?
There is. There's definitely both a career development aspect, which we actually do spend a lot of time thinking about that, how to encourage people to grow and giving them opportunities, whether it's, to learn communications training or, to learn a new skill. But also there is a lot of guidance that's given to try to help people, get to wherever they ultimately want to go. There's also, of course, people have a job to do, and that's the majority of why they're here. But the career development definitely matters. Also, mentorship doesn't end when somebody moves to industry, and I still keep in touch with a lot of colleagues and with people who I very much value in academia. I also now have a executive coach, which is a new thing for me, who's working specifically with me. I meet with her every 1 to 2 weeks. This was something
I want that job. I want to be the executive coach.
Yeah, it's interesting because it's different from like a therapist or something, although it is very highly personal, but it's really about helping you get from where you are to where you want to go, and developing whatever skill set that you want to develop. They begin with a 360 review of people that report to you, people you report to, and colleagues that are at a similar level to you. And a good 360 review, it draws a little blood. It doesn't require transfusion, but it's helpful to see yourself how others see you. And so that process is going on right now.
The episode I aired of the podcast this week was with a life coach, was a GI pathologist. Her name is
Christina
Arnold, right? Yeah. She left GI pathology and she's a life coach. Take a listen to it. I was fascinated. I learned so much about this. I started Googling life coaching schools. I'm like, I want to be a life coach too. I can do it. It's interesting.
This is an executive coach is like a life coach, but specifically for people who are in leadership positions in the corporate world.
Yeah,
because there's certain skill sets.
Yeah, Ross, do you believe in peer mentorship? And what I mean by that is there a point in your career where you don't need to have a mentor? I mean, when you're a professor and you're running this, or do you always feel mentorship is a continuum, that it doesn't mean you're never above being mentored by your colleagues? Maybe it's not mentorship, maybe it is advice, whatever it is. What are your thoughts about peer mentorship? I call it peer mentorship.
Yeah, I mean, I think you're never above it. When I have to navigate things, like when I have had a few opportunities in academia to think either about new roles at Sloan or opportunities elsewhere or choices I want to make about my science, I've sort of fashioned a sort of peer group of folks that I think we're all helpful to each other as we navigate these things. One of whom is David's boss, Jay Bradner, who's my college classmate, and a number of others. And I think we all go to each other. When I was making recent important career decisions, I even ran the choices and job descriptions by folks and talked through it and went and got some advice. And it's funny, I was talking to one of my bosses at Sloan who said to me, you need to go and get some outside advice about what you're going to do and whether you're going to be here or not. And I went and talked to some folks outside who I viewed as having experience I didn't have on things. And I just needed to talk through things. And it was immensely helpful for me. The other thing for me, I think, is that, and I grew up, at MGH and then Dana-Farber. And I was very lucky, and many of us are, but my peer group that I did my medical residency and then fellowship with and started in the lab includes some pretty extraordinary people. It includes people like Ben Ebert, who David mentioned before. It includes Jay Bradner. It includes Levi Garraway, who's the chief medical officer at Genentech, Jeff Engelman, many others. And we kind of grew up together. And whether we're at academia, industry, I think we all sort of got a lot of back and forth with each other. And I felt, and again, it's like everything else in medicine, like these are the people I took call with when I was an intern in the unit, right? And then they're going through the same career like journey as you 20 years later. It's like you went to war with someone, and those people are immensely valuable to me. And I'm very fortunate because some of them are extraordinarily thoughtful. Obviously, they're very productive, and they're always just a text or a email or a phone call away. And we can maybe get to meetings. For me at my stage, the primary thing I get out of meetings is, or one of the two things I think that are important for me that I get out of meetings is catching up with those groups if they're not local. Like when I go to AACR, seeing my posse is really important to me, much more important than going to a large event. The other thing for me is meeting young people that I might not otherwise meet. The rest of meetings, and maybe we get that, I could do without.
That is literally perfect segue. It was not rehearsed about the meetings. I mean, maybe David, do we have a lot of medical meetings? It seems to me that every weekend you could literally be in a meeting somewhere.
Yeah, we have too many meetings, certainly.
Why? I think it's economics. They want to make money.
Well, it's a little like why we now have too many classifications for hematologic diseases. I think there's a certain ego that goes into setting up meetings, but there's also, people want to discuss specific topics, and there are different groups, the folks who go to ASCO, who go to ASH, who go to AACR, who go to EHA, overlap a little, but not a lot. And so that's part of it. And then you get to all the smaller disease specific meetings, and it's very different.
David, you've got ASCO GI, ASCO GU,
Yeah,
SGO, Society of Gynae Oncology, AACR, ASCO, EHA, World GI, World Lung, ESMO GI, ESMO Lung, ESMO Asia, ESMO Latin America, best of ASCO across the year. Then you've got ESMO, ASH, SITC, Society of Neuro-Oncology. I can probably literally recite about 25 meetings without even looking at them.
yeah.
Ross, what do you think? I mean,
Way too many. Way too many. Maybe logarithmically so. I think that they've been good for academics who want to get their name out, and it's easy to get on the meeting trail and view it as an end and not just a means to an end. And that's a dangerous slippery slope that I know at points in my career. It was great to be on the circuit, and it becomes its own thing versus really asking yourself, well, why am I going to present our science? Is it because I want to share something, or have I given that talk 8 times before? And I think that it's easy to give that talk again, and it's silly to do that. That's one aspect. I think the other aspect is that I think, many of us in academic medicine, whether lab or clinical, we spent the first 35 years of our lives like sweating in hospitals, never traveling. And there isn't a part of it where people want to do things. And I respect that. I don't mean that in any pejorative way. I will say I have had the blessing of having the chance to see a lot of the world with dear friends in wonderful places, and I enjoyed it at times, but obviously I have different perspectives on it now. The thing that I feel strongly about is that, you know, some of this you can control and some of it you can't. And the way I view it is the following, is that if you, I can give the same talk on leukemia genetics and epigenetics, you know, 15 times a year if I want at meetings. And I did it. I'm not doing it anymore. And I realize I'm in a privileged space, that I have stature, but I'll pick the 2 or 3 that I'm excited about and where I think I've got something new to share. And then I'm going to suggest other people for the other ones as best I can, because I think those opportunities are people that really deserve and maybe don't get those opportunities.
But Ross,
and I think that's,
do you think the first 5 years of your career as a clinician, scientist, or researcher, you kind of have to say yes to everything so people get?
I think it is. I think you do. There's a, I hate to use the word, you're building your brand, but you do want to have the chance to present your science in the rooms where the people that review your grants and papers are. You want to visit institutions where those same people are. You want to build your collaborations. And I definitely encourage the people from my group that starts their lab for the first 5 years. But I do think, as a lab investigator, for example, I tell my people, don't go to ASCO. Don't go to, I get that they may want you to give that heme talk, but that's actually not going to solve what I just described.
Yeah.
What you want to do if you're a lab person is give a talk at a scientific meeting where the people who review your grants and papers are. You know, every 3 years you want to go to ASCO, so be it, fine. But don't do it. Like I don't go to ASCO. Like,
so what meetings do you go to?
So I go to ASH every year, although I have this dream in the next few years of doing an ASH hiatus when I'm at some point where I stay home, maybe when I'm done with my institution, my ASH responsibilities. I may get in trouble for saying that, but I have a dream that one year I'm going to send photos of all the great places I am with my family. Everyone's at ASCO. I did that for ASH. I mean, I did that for ASCO this year, but you know, I go to ASH every year. I have probably 3 or 4 core meetings in my field. There's a FACET heme malignancies meeting. There's a Keystone hematopoiesis, and that I do. And those are important to me because those are like a recurring meeting where my people, like my field, go. And it's important for me to be there to hear their science and to show our science and to network. And then I run the AACR course for fellows who are going into the lab. That's in a couple of weeks in Aspen. It's the lab version of Vail. And that's very important to me, to run a course to help people get started. And I do that. The rest I do is by choice. And I will tell you, and David and I have spent a lot of time talking about this on and offline. I'm mostly choosing to not do it. I've got a travel stretch from late July to mid-August of a few meetings. And then, you know, I think I'm giving, I've got one meeting at ASH headquarters in September, and then I've got one lecture in October, and that's it. And again, I live in a privileged world. I also live in a world where people ask me to give Zoom talks where I can do it from my room. I also live in a world when there's a hot story in science that maybe people don't hear about. I'm often getting to review it as a grant or a paper. So I use Twitter and all these things, which I think are useful. So I realize that. So I do encourage people early in their career, or if you're not at a big institution, you got to get out there. But then if you actually start to feel like you're achieving and people know who you are, you should use that stature as a way not to keep going to meeting and to be more selective and to use that privilege as a way to pick your spots and to advocate for others to get the opportunities that maybe you don't think you need anymore. You need both those things.
David, tell us about some hiatus story pertaining, I mean, because listening to you, you've been on the travel circuit. So 2 questions for you. Number 1, travel in industry is a little bit different than travel in academia because you probably have responsibilities to travel for business and other things. So how is that shaping up? And do you have the leisure to do what Ross is doing, which is he can say no?
Yeah, so I've been thinking a lot about this lately, actually, Chadi, you know, partly because next week, my daughter, who's a PhD student in molecular plant sciences, working on single cell algae extremophiles, is going to her first meeting at which she'll present. She's going to this international symposium on carbon utilization by photosynthetic organisms at Princeton and giving a talk. And so now it's the baton is being passed to the next generation. Tatiana Prowl, who was going to join us on this podcast, but FDA wouldn't let her. She and I have become friends over the years and have had a number of good talks about meetings and COVID. She's very concerned about COVID safety, understandably, with meetings, and about carbon footprint. 3 to 4% of the current global CO2 output is air travel, and that's the predominant way of getting to meetings. It's great to be talking about this with Ross too, because we have talked about this several times over the years. One of the most fun trip memories I have is of a small meeting in Dubrovnik, Croatia, organized by Serge Verstovsek from MD Anderson and some of his Croatian colleagues. And Dubrovnik has this fantastic medieval wall that you can walk around. And
King's Landing from Game of Thrones.
It is. Well, this was pre-Game of Thrones, but now everybody would recognize it who's seen Game of Thrones. And we went and walked around together and just had a great conversation. It was a wonderful day. I got to know Ross a little bit better. And those are the kind of things with an in-person meeting that happen, and you make a friendship or strengthen a friendship.
Yep.
that can last for the rest of your life. That said, in 2019, I think Ross and I were probably both just getting a little burned out on travel and challenged each other to take a 6-month hiatus from travel starting in 2020. And the dates we picked were sort of late spring to fall when things got busy again. Well, and then COVID happened, and that travel hiatus became mandatory.
So you're behind COVID.
It was unexpected, but it was wonderful in some ways. I took up woodworking. It was great to be at home, just a much more relaxed pace and quality of life. On the other hand, I miss people. And when I went to the ASH annual meeting in December, which was the first meeting since March 2020, everybody I talked to, just about 1 or 2 sentences in, and people would tear up, or they'd give you a second hug. They were just so happy to see each other again and to be able to relate what was really going on in their lives. Because it was also, in addition to not being able to travel, of course, we've all been through tremendous trauma in different ways, some more than others in the last 2 years.
Two things Go ahead. I just want to add, Dave, 2 things. One is that one of the things I'm proudest of for 2022 so far, and I think scheduled, is that I'm going to travel as much for my daughter's ice hockey as I will for work, relatively similarly. And that's a goal I have, to travel as much with my family as I do for work. It may not get there, but certainly that's something I've thought a lot about, is that I definitely want to make sure that I'm more balanced and when I do travel, and that I do that. And the other, I will tell you, it's funny. David gives one version. I actually had a very different experience. I was at ASH this past year, like him. It was my first away meeting. I'd only been in a hotel when I was with my family, either on a drive vacation or hockey tournament. I had not done work travel overnight. I'd done a day trip to Boston in November of 20 as a dry run. I actually was miserable there, to the point that I went home early. And I will tell you that, and I'm an extrovert at heart, I found that during the pandemic, I developed a real aversion to the large parts of meetings, like the big room, the big reception. And I'm sure some of it was the sense that Omicron was coming, and the sense that even with masks, it just felt like you could never get rid of the risk. But it's funny, I'm struggling with that, and I'm being really honest about it.
But Russ, is it safety, or is it just because you feel no value?
I think that it's that I developed, I got out of that getting used to being in water, and I found that I don't enjoy it the same way I used to being in the big room. And I'm always like, why am I here? And so what I learned and I've done a few meetings a day, CR, was that I go to the big rooms when I need to give my talk, whatever. But in general, when I go to big meetings now, I schedule mostly smaller events with small groups. I'm much happier at ASH or ACR at a dinner with 3 or 4 friends, and that I am and I avoid the large receptions, and that's very different than I used to. I used to love going and shaking, and maybe it'll change, but I have to say, I think for me at least, it'll be interesting. I'm not sure. I don't know. Like, let's see where we are in December in New Orleans. I think whether I go to the big events or not.
Do you have that luxury, David? I mean, I think I'm going to assume that you probably don't have as much freedom in making these decisions like Ross.
Yeah. I do have some control, but one of the things that we're doing at meetings is meeting with investigators and meeting with other companies, particularly companies where we may partner with them or where we're considering buying them. And so that aspect of it is I think, going to be with me to stay as long as I'm in this role. I had a very similar reaction to how Ross described ASH at ASCO. I went the first day, it was overwhelming. It was so much fuller than ASCO, or than ASH had been. There were over 30,000 people compared to, I don't remember what ASH was.
There were about 10 at ASH this year.
Yeah, it was a third of that, right? So, and I went into a lymphoid malignancy session. Every chair was full. Almost no one had masks on. Like every single chair, like all and I just got this like weird claustrophobia sense. And I tried to stand in the back. And of course, the guy from the McCormick Center came over and said, "You can't stand here, fire marshal rules." So I said, okay, maybe it was just this session was packed. There was some good new data being presented. So I went to another session a couple hours later, same thing. I mean, even though I was there a few minutes early, there was no chairs. It was totally packed. And I said, enough of this. So I watched the rest of the meeting, the scientific part of the meeting, in my hotel
in your room. Yeah,
I had a nice view of the Chicago River. It was lovely. And I went to all the individual, I met with Guillermo Garcia Manero from MD Anderson and his colleagues. And I had a meal with Ruben.
But again,
Mesa,
David, the same question to you, take COVID out of the equation completely.
Yeah.
Is that, I guess I'm trying to separate the feeling about the meetings, how much of this you're afraid of catching something, right, versus you has nothing to do with COVID. You just feel, I've grown out of this.
Yeah, I think it's a little bit hard to disentangle them right now, given that we're just reemerging from COVID. That said, I love being able to be in the room and watch a talk that was just given at 1.5 speed and not have to, fight the crowds. And if something isn't what I expected, and you're the fourth seat in, to have to disturb everybody to come out of the aisle and go somewhere else. And, and to be able, ASH always had simultaneous sessions where there were multiple things you wanted to go to at the same time. Now that's not an issue because you can watch the things you want. I'll go if at ASH, it was always really important to me if I had like a trainee that I'd worked with to, be there in the front row to make your presence known and support them. But it's different now. I mean, we have presentations that are trials or studies that we sponsor, but it's not the same as it was when I was at the Farber.
I went from going to meetings and then watching my daughter's hockey games on simulcasts from wherever I was to being at my daughter's hockey games, watching the meetings and simulcasts, and I will take the latter over the former. And that's a very personal choice.
Yeah, I mean, yeah, absolutely. Well, this has been amazing. I mean, time just flew by. I love this. I think we've covered mentorship, mentees. We've talked about the career path. We've talked about meetings. Before I let you go, and any parting thoughts, David, we'll start with you. Any last comments? Anything we probably should have talked about that I did not capture?
Well, it's always fun to talk to you, Chadi. We covered a wide range of topics, and I always feel like when I talk to Ross and hang up the phone or, dinner ends, that I could talk to the guy for another 6 hours because he just has so much energy and insight. And so I'm particularly honored to be on here with Ross. I've really enjoyed our friendship and hope I see you, if not at a meeting, some other fun place.
I'll be at a hockey tournament mid-August in the Boston area. I'll ping you. Go. I will see you in Boston.
Let's hope New Orleans. Yeah. Ross, any parting thoughts? Anything maybe we should have covered that didn't catch?
No, this was great. I hope I know there's many more to cover, and I hope it's not my last time here. I promise to come back if you'll have me. And David, it's always great to see you, my friend. And Chadi, I will say it's been just a pleasure to get to meet you virtually, and I can't wait to meet you in person and to have some Skarbnik barbecue together.
I am looking forward to it. David?
One last thing, this is, you say New Orleans. This happened last year with the Georgia voting law. There was a call to move ASH. I was kind of against it because, it's always the service workers who get screwed the most, and, they depend on meetings and such. This year, the Louisiana issue is a little bit more complicated because it's a safety issue with the Roe v.
trigger
Wade
ban
being struck down. And so this may be more than just politics. It may be safety of attendees. And that, to me, is in a different category. It's also a little hypocritical, I guess, because we have in the past gone to meetings in countries where, the safety is not guaranteed. That said, this is a really, something that I'm really wrestling with. And I know ASH is and other medical societies as well.
I'm not going to say more because I have a role with ASH. So I'm going to defer to the societies we try to navigate the issues David just brought up.
Yeah, I wasn't aware that ASH was looking into this. I serve on the ASH quality subcommittee, and but I didn't know that this is being discussed. Maybe, but I'm sure they will let us know at some point.
We'll hear. Yeah, we'll hear at some point.
Guys, thank you so much for coming to my modest podcast. If Tatiana is listening, she missed out on a fun podcast.
Yeah, she totally did.
Totally.
I'm going to make her feel bad.
Missed her.
Totally did. You have to make her feel bad. All right, guys.
I will.
Thank you so much.
Okay, bye.